Okay, hi, this is Miss Litton, and these are my wonderful AP biologists. Say hi.
Hi.
And we're getting ready for part 1, our essays tomorrow. Let's take a little business first. What is, okay, don't type so loud that you annoy me. You know who you are. Okay, first thing, you need to make sure that your plants are in the right spot before you leave today. For some reason, we have 2 period 1 group 3 controls. Can only have one. We had, now I'm going to say there could be a chance of some user error because the period 1 group 3 light was over in the misty area. So I took it out of the mist and I reached to put it into the light.
There's light there too.
So now we know who put it in the wrong spot. Thank you for that clarification. We wondered. Okay, it's all clear. Anyway, per your group, you should have 2 plants. Why don't you double check before you leave today and make sure they're in the right spots so they spend their first 24 hours in the right spot. Number 2, don't talk when I'm talking because it's annoying and you can leave. You don't have to be here. Number 2, your lab. Your pre-lab is due tomorrow, right? Hopefully you didn't wait till today to do it. That would not, hashtag not smart, okay? You would want to, your hair looks super cute today. You want to go all the way up through the data tables, right? And I got a really good email over the weekend asking me, Miss Litton, for the part 1 when we count the stomata, should we use that equation that we are going to have to use to calculate the number of stomata? Yes, that would be an equation that you're using some math. You could throw that in there, so good job. Yeah, I think that's it. Are there any questions just on the lab that you need me to address? Yes?
Going for that flow chart for part 1, there doesn't have to be key steps, right?
No, just like a picture, because it's really more of an observation, but you just want to use the equation that you used during that observation. Okay? All right, anything else in the lab? I have gotten the same question, I want to say twice, so in case you haven't got this part yet, when you write your hypothesis, yes, you could write it specifically for your variable, but since you're addressing transpiration on the whole, you might want to write a hypothesis about how the environment impacts transpiration in general, right? And you're just putting your plants into different environments, right? It says that in the instructions, but I had somebody ask me about it again today. Anything else on that part of class? Okay, second thing I want to tell you is when we come into class, first thing we're going to do is get that done and do our lab, you know, check your lab notebooks. Then you're going to do your stomatal counts when we start class. Then the last 40 minutes you have, which is really too long for what you're doing, right? Because you don't even have 40 minutes when you answer the full every chapter, right? So that's really enough time, should be last 40 minutes. I'm going to tell you I spent, I want to say 4 or 5 hours working on that all yesterday morning, writing your questions out and which ones you were going to get. Maybe I had a couple cups of coffee in between there, but I think they're good questions. And there are, if you think about it, the essay part is, your essay is always worth 50%, your multiple choice is always worth 50%, right? So the full multiple choice will be when we do the whole test, right, 50%. Then you're doing part of the essay now and a little bit of the essay next time, just to kind of break it up a little bit. So percentage-wise, 35 of the 50 points are tomorrow. 35 of the 50 points are tomorrow. Okay? So breathe. Okay? I asked each class 6 questions, and then the points are skewed between them. Do you want to know some for sure's that I asked you?
Yes.
Okay, this is a gift. This is a gift that I'm giving you. We're going to pause because if you're not here, you're not worthy of hearing. Just. I'm kidding. I'm sure you're doing something really important. Okay. Okay, I'm kidding. Okay. Don't pause. And then you're all like, okay. First thing I would for sure know, I bet you could have predicted this, something about life cycles of viruses. She's like, they're just, remember life cycles. What are the 2 cycles? Lytic and life cycle. Do I expect you to draw it?
Yeah.
Okay. A picture that I understand that matches it?
Mm-hmm. Yes.
Okay, so this is a gift I'm giving you. Those ought to be perfect. You ought to be able to make sure you can name every step, that you have a picture for every step, that you remember things like where would you put prophage, right? All those kinds of things. So make sure you can do that. Like with the bacteriophage, which is really obvious. I can see its structure, I can see its parts, I know what it looks like. Does that make sense to you? Okay, that's a for sure, right there. Would you like another for sure?
Yes.
This is me being very loving to you because I'm sure you have a very busy week and you're stressed out. Your second for sure is something about plants.
Oh, great.
Any guesses?
Alternative
Yes.
Oh, I know that. I'm sure that. Yes, sorry.
I love alternative generations. I can throw it.
Another for sure. Another one. Okay. Now. Yeah.
Would that include like the macro and micro?
Humidified?
Yes.
No. You can make it be homosporous. Yes.
So those are for sure questions that are going to be on our test tomorrow.
Let's see, how do I make that more clear? Like I'm Nancy Pelosi, no money for the wall. Yes, those will be on the test. Okay, all right. Sorry, yeah, I just see that press conference. Okay, next. It's not political. Okay, are we good on that? Okay, now, would you like some suggestions?
Yes.
I gave you all the essays. You could look through those, right? Make sure. Potential essays you could do. Here's another source of potential essays. When you go through the chapter, because I know you don't have time to do everything like at this point, right? If you haven't studied yet, it'd be like, ugh, okay? So you need to like focus your energies. Have you ever noticed that there are questions embedded in the chapter as you go along? You might try those. I'm just saying. They look like an essay, you might try those. It's crazy. Okay? You know where else? Is at the back of the chapter. Each chapter, there's questions there. And yet, here's another thing. At the end of the notes, I write what you must know. They're written as statements. Maybe you should know those. Okay? Now, if you have a good friend and you have time, if you tag team and go back and forth on that and review them and take the lead and review each one of them, that might be something if you have a really good friend or a good group that you can do. Because I can't lecture on 1, 2, 3, 4, 5 chapters in this amount of time, but I did give you a good head start. Okay? Things about origin and history of life, that sounds like somebody's going to get a question on that, if not multiple groups would get a question on origin of history of life. What are some things I could ask you about origin and history of life? The 4 steps. How about that? How about taking care of all the obvious ones? When I give you, hey, 4 steps. Here are the 4 things you need to get onto land if you're a plant. You know, those obvious things, know those. Like chapter titles and things like that. Review those, okay? Go over that part. Something like the geologic time scale. That would be really important, right? Knowing Precambrian, Paleozoic, Mesozoic, Cenozoic. Knowing the years, right? And what are the key things that happened? When you talk about, let's say, the Paleozoic era, animal-wise, where did you start? Paleozoic, animal-wise, invertebrates. Where did you end? Could you name all the way in between, maybe all the other ones? Because that's what I would do if I wanted to get 100% on it. I wouldn't just give me the bookends. I would tell me what happened along the way. Does that make sense? I would know the years. I would know the, all of that. Okay. Chapter 19 was about phylogeny. I would understand cladograms. I would understand the point of my blast lab. What were you doing in the blast lab? That's last week, I can't remember. Okay, think about that blast lab. What did you do in that? What is classification categories? Remember those? Domain, kingdom, phylum, class, order, family, genus, species. How did we set those up? What are the origins of that? What does it mean to be in the same phylum? What does it mean to be in the same class? How do you determine that? Right? Those are all that's like big ticket. What else is that? Knowing the domain, right? All of that. Yes. Yeah, raise your hand.
Oh, I was just going to say I know the different domains.
Yes. Okay. Am I still recording or did I miss it? Oh, no, we're still going. Okay. Viruses, we already talked about some key things about viruses. and bacteria and archaea. But remember, one of our big tickets of our 4 big ideas is evolution, right? How could viruses evolve? How could bacteria evolve? How could they get new traits? You know the life cycles of bacteria. You know the life cycles of viruses. How could those change over time? Where would we see evolution? Why do I have to keep getting a flu shot every year? Bacteria, archaea, differentiate between them. And then that brings us to chapter 23 and chapter 24, plant evolution and structure. On plant evolution, I mentioned this before. What are the big steps that you need to go through in order to be a plant, right? What steps do you have to go through to get on land? Why would you even want to be on land? What's the advantage of getting on land? And then structure. We study, what are some major plant structures? Okay, so vascular tissue that are found in what plant structures? Roots, stems, leaves, those are plant structures, right? Remember we looked at modifications of roots, things they could have on them? What are some things they could have on them? Root hairs. What's in the stems? What is their main component that they have inside of them? Xylem and phloem. Okay. All right. Leaves. What's the job of leaves? Photosynthesis. Why do they have holes in them? Right? Yeah. So what I'm asking you to do is look at these different plant structures. You learned about them. What is the point of them? To go big ticket items. What are some of the adaptations they have that make them suited for what their job is? How does structure equal function? When we start going into organisms and we transition into human systems, we're going to want, why is this structure really good for this function? For instance, if I think of a structure on roots, what would be something? There are multiple structures you could come up with on roots, but just give me one of them. Okay, prop root is a type of a root. A structure on a root. Root nodules. Okay, let's go with root nodules. Sure. What's the point of root nodules? Store what? Cyanobacteria. Cyanobacteria, that's something to know about. Have we not talked about that endlessly? What's the importance of cyanobacteria? How has cyanobacteria affected our earth? But that is a structure. A root nodule is there to hold cyanobacteria so that the cyanobacteria does what? Fixes nitrogen for the plant. You said root hairs. What's the point of a root hair?
Increased surface area.
Yes. See, structure, function, that's what I'm talking about. Some of you are doing all the memorization and forgetting what the story is even about. Okay, so make sure you understand the big ticket. Plant evolution structure. There's something else I was thinking about that. Cyanobacteria. Oh, the different types of cells that you would find in a plant, those would always be important, right? Apical meristem and what tissues are derived from it. Last but not least, my friends, do you think I'm going to have the highly suggested reading and thinking on there?
Yes.
I am. So it'd be a good idea to kind of read through that as well. Right? If you've already read and studied for each quiz, that should be easy peasy. Just look through it one more time, right? But if you never invested and you just read other people's work, then you know it's going to be harder for you. Just accept that. Okay, I'm now open for general questions. Yes, I do love you, but go ahead.
This is the about the format you mentioned in the beginning. You said like there's going to be 6 questions. Is that in both essays, like today and in 2 weeks, or just
No.
tomorrow?
I only wrote tomorrow's essay.
Okay. Oh, thank you.
There will be fewer. Actually, I kind of, because I needed to look at the whole unit, I'm looking at about 3 essays for the next one worth 5 points each, or somewhere in there, you know, for 15 points. I actually did start working on that. What else? Stranger danger. Sorry. Anything else, you guys, that you want to talk about? Now is the time to ask, not huddling around me at the end because then I'm going to say, oh, we're done now. Okay. Yes?
Can you kind of review the differences between the animal virus and retrovirus?
Sure. So typical of an animal virus is that because animal cells have a cell what? Membrane and no what? Cell wall. So what happens is they are typically, doesn't have to be, but they're typically engulfed by the host cell. So where you saw bacteriophage on bacterium, and it's got to get to the cell wall and the cell membrane, then you're going to see that penetration where it might be injecting its nuclear material in. Animal cells, they tend to hide, because bacteria can be everywhere, but an animal is one unit, and so if you're a virus in there and you want to hide, so they get engulfed because they've been traveling around with a cell, like a type of membrane around it. So they get engulfed, that membrane has to come off, the capsid has to come off before you can get to biosynthesis. What is unique about a retrovirus in all of this is its viral code, its genetic code is RNA. And it has the ability to get into the host cell's DNA by using the host cell's machinery to make a DNA copy of itself. So it uses that reverse transcriptase to make a DNA copy of the RNA. Another enzyme that gets formed is replicase, make it double-stranded. Another enzyme is integrase, which gets it integrated into the host chromosome. Once it goes into the host chromosome, when it comes out, it could always take a little bit of the host chromosome with it, right? Or it could have brought some other host DNA with it as well, which could cause change over time. And then when they go out, they don't tend to lyse the cell. They could, but they can lyse the cell, or they can do that where it puts a membrane around it, kind of oozing out. Yes, question?
Retroviruses. So they're the ones that are hiding in the DNA?
Yes.
Okay.
Mm-hmm. Yes.
You remember stage 4 with biological evolution?
Yes. Okay, so what she's talking about, let's just back up and make sure we got the other stages. Stage 1 was the formation of monomers. Good. Monomers, organic monomers. Good. What was stage 2? Polymers. What was stage 3?
Protocells.
Protocells. Stage 4 is you are self-replicating. At this point, you're not just an entity floating around, but you have some sort of genetic code of some sort of information, and you're able to make other cells. So you're able to replicate. That's stage 4. Hey, what else? Yes?
The classifications, can we go over what the human...
Sure. What domain are we in? Eukaryote. We're going to remember that because there's only 3 domains. You know we're not bacteria and archaea. That's how you remember that. Domain. Kingdom. I always think animal kingdom. Domain, kingdom, phylum. Chordata. Now sometimes some people are going to want to say vertebrata. That's actually a subphylum. Okay, so you won't use that. Okay, chordata. Domain, kingdom, phylum, class. What is it? Mammalia. Look at all the mammals in the classroom. I'm looking at mammals. Okay, with mammalian-like characteristics. I see all your hair. Domain, kingdom, phylum, class, order. And I always remember, no monkey. And so the name of that monkey we're going to say, yeah, it's just fun to say no monkey. That's why I like to say it. But you're going to think about primata. Domain, kingdom, phylum, class, order, family. The family is Hominidae. Hominidae. And then you're going to remember Homo sapien, genus, species. Now, it's kind of a misnomer because when you give the species name, you have to give the genus name too. It doesn't stand alone. It's like a companion cell with phloem. It does not stand alone. Yes?
When you're listing all of them, do you have to list the subspecies sapiens, the Homo sapiens sapiens?
No, but you'll know that because you're smart. Okay, what else? Yes?
I'm just making sure if you want science structure, you want broad descriptions, or do you want very detailed to each part?
If I asked for something on that, I would make sure you know the obvious things like roots, stems, and leaves, right? Those are the big plant structures. Those are the ones we talked about.
Yeah.
Now they have components that you would want to be familiar with. Monocot, eudicot, that's something else you could review, right? Yes?
Can we go over the primordial soup hypothesis?
Yeah. So the idea there, this would be in the step 1, right? And there's a couple different ones. There's the extraterrestrial, right? Came from somewhere else. There's iron sulfur world. We got everything we needed. And that's used for several of the steps, right? But the one that probably is most likely, and that's the one I would always use, is that the gases on the early Earth were ones that probably came out of a volcano. And the early Earth had no free oxygen. There's nothing to generate free oxygen. There was oxygen with H2O or carbon dioxide, but there wasn't free oxygen. So this early Earth had gases out of a volcano, and it was a reducing environment because there was no oxygen. As a result of that, it was very conducive to building, not breaking down as oxygen does now on our planet and our Earth, right? That's why apples turn brown. When you take a bite out of it and you haven't taken a bite in a little bit, it turns brown because oxygen is interacting with the apple. What kind of cells in the apple? Parenchyma, okay? And it's oxidizing, right? Parenchyma, right? What are the other two? Parenchyma and parenchyma. Parenchyma is alive, right? Okay, so anyway, back to your question. So early Earth had these gases out of a volcano. It was a very reducing environment, electrons getting donated left and right, and we had a source of energy. The source of energy could be
Light from the sun.
UV radiation, light from the sun, because there was no what layer yet? No ozone layer, so it's full-on sun, UV, everything on the electromagnetic spectrum, basically. You had lightning, you had molten magma, and you had radiation, right? Decomposing isotopes. Okay, guys, you might understand that, but somebody asked me that question, so give them a respect of being able to hear me, right? That's fair. Okay? Yes. Wait, you just asked a question. I got to go with somebody new. Who hasn't had a question? Yes.
I'm a little confused on meristematic.
Meristematic tissue. Anything with stem. If it has stem in it, like meristematic, that tissue is capable of dividing and making more tissues. So we have bone marrow that can make more cells for us, like blood cells, right? Plant tissues have apical meristems, which are the tops of the shoots and the bottom of the roots to always make more shoots and roots. And that's pretty much it for monocots. And that's why they grow straight up and straight down. But eudicots have what's called vascular cambium, which is still a meristematic tissue. And it's able to generate more xylem to the interior of a plant and more phloem to the exterior of the plant. And that allows for branching and increase in girth as well as size. Yes?
When you said to know the steps of plants getting onto land,
Yes.
do you mean the adaptations to land, or?
Like when we did them on the cladograms, like what was, remember the different things we talked about? There, and I had a slide and had the pipes on it and seeds and some flowers on it. Remember all of that? That part. Yes. No, I'll let you go over. I gave you enough. Yes.
What are like the root cell zones, like the elongation, cell division, what are those?
What about it? Be more specific, sweetheart.
Like, can we just go over
The 3 zones?
Yeah.
Okay, is that something you could reference in your notes? Zone of cell division, zone of elongation, and zone of maturation. Those were the 3 zones. Which one had the root hairs? Maturation. Because if you're dividing and elongating and you've got a root hair attached, it could get ripped off, right? Yes.
In the notes, in like for chapter 18 in stage 4, it talks about the protein first and RNA first hypotheses.
Yes.
What is the relevance of like those hypotheses in stage 4?
Okay. All of the stages have hypotheses that they use to support what happened next, right? So it's kind of like you go, what would have had to happen first? Well, we need some organic molecules. Well, how could we have gotten those? So one idea is the whole, organic soup, volcano, hydrothermal vents. If I was going to talk about it, I'd do the organic soup because that's the most widely accepted. Okay? Then how would you get polymers? And which polymers came first? And we know the 4 important organic molecules are what? Carbohydrates, proteins, lipids, and nucleic acids. So one argument of those 4, which are critical? Proteins and nucleic acids, because proteins act like enzymes, and nucleic acids have our genetic code, yes? So, of those, one might say, well, I bet RNA came first, another one might say protein came first, another hypothesis would say they happened at the same time. Remember Karen Smith and the clay, they happened at the same time. Another hypothesis would be that RNA came first because it can act as both a substrate and enzyme. Another one would say proteinoids, because they're so easy to form, maybe they came first. That's what that's referring to. Yes?
I just want to make sure that I understand this regarding the question for hypothesis. A
Pressure flow hypothesis is not on your test.
Okay.
We didn't even finish talking about it yet.
Okay.
Yeah.
Can you go over transformation, conjugation, and transduction?
Okay, so here's what I'm here for. I'm here to make known to you something that's unclear. If there's a definition that you just need, you could look that up, right? Is there a part of those three you don't understand?
I don't understand what transduction is.
Okay, let's talk about transduction. Transduction is when a virus takes DNA from host 1 to host 2. Is it doing it on purpose? Probably not. It's just a factor of if you're in, like a bacterium especially, right? They don't have a nucleus, they don't have sealed off DNA inside of a nucleus. So if you're a virus and you go into a bacteriophage, that genetic code's right out there. It's possible that you pick up some of the DNA that's in bacterium 1 and make it part of your viral genetic code now. So when you go into bacterium number 2, you took some of bacteria 1's DNA with you. It wasn't yours, you brought it from the last host. So that would be one way to get new traits into that bacterium. Does that make sense now? Yes?
Can you go over Pasteur's experiment and his conclusion?
Pasteur disproves spontaneous generation, right? And so this would be one I would say is a lookup, not me explained. But he did spontaneous generation, disproved that. And he did that because people used to think that life could come from non-life. They would see maggots crawling out of rotten meat and say, that's where maggots come from. Maggots come from rotten meat. What they couldn't see was the fly's eggs because those were microscopic, and so nobody saw that. So what he did is he said, okay, see this soup, and yes, there's stuff growing in it when I leave it out on the counter for 2 or 3 weeks. What he said is, I am going to boil it, kill off any life is in there, and I'm going to cap it off. You're not going to see any life because I've destroyed it all. Life can't come from no life. But then people of the day said, you took away one of life's vital forces, and that is air. Okay, but you and I know little things are in the air, right? That's why you should cover your mouth when you sneeze. You're putting all those particles out to us, right? So what he did is he took a flask, boiled it, but he had the flask built so it was a swan neck flask. So anything, air could get in and out, a vital force, but any bacterium or any particles in the air got trapped in the bottom curve of that swan neck, and his fluid inside stayed clear. Does that make sense? Yeah. Okay, what else? Yes?
Since the pressure flow hypothesis is not going to be in the test, is there a possibility of the TAPC theory being in the test?
Well, if you remember on the calendar, I have exactly what chapters are on the test.
Okay.
So that tells you what chapters you're accountable for this test.
Okay.
Okay. What else? Anything else you'd like me to go over? Any other questions? Yes.
It talks about, like, in the lytic and lysogenic cycles.
Yes.
It talks about how, under the second one, how...
I don't know what second one
Sorry,
is.
lysogenic cycle.
Yes.
It talks about how, like, environmental factors can, like, push it back into the lytic cycle.
Yes.
But wouldn't it be going back anyways?
Well, no, what it's saying is, why does it come out of hiding? What triggers it to come out and re-enter the lytic cycle? Yeah, so if the conditions of the host change, then it would be to its advantage, evolutionarily speaking, to get out of that host. So now's the time, prime time, for you to start making more viruses and release them, because something might be happening to your host.
And just to clarify, in the lytic cycle, it's just going, like, it doesn't
Going
try to hide.
in, taking over, right? Going through AP biomist or attachment, penetration, biosynthesis, maturation, release. What's the lytic cycle happens is you still have that, but you took an off-road, right? Off-road, right? You took a side road and went, and you are doing integration, and you're integrating yourself into the host chromosome, and you're going to lay latent there as a prophage. And every time that host replicates, you get replicated right along with all the other DNA because you're hiding literally in its DNA. When something happens to that host and the environment changes, that's when they would leave the integration stage and go into the biosynthesis stage.
Thank you.
Okay? Anything else? Yes?
How exactly does the active transport work for
That's not on your test. Yeah, look at the chapters on the list. Yeah, I'm happy to talk to you about that because we were mid-discussing that last class, but it's not on your test. So do you really want me to explain it right now? Okay, yes.
Talking about gram-positive and gram-negative, when it says gram-positive is thick-walled, purple, and trap stain, what does the trap stain part mean?
It's because there's no additional layer. Gram-negative has a little bit thinner wall, and they have another layer, like another cell membrane on the outside, so 2 membranes. So when you're gram-positive, you don't have that membrane, your wall's thick, and the stain can soak into it. Whereas when you're gram-negative, you have the wall, and then it's like you have a baggie on top of it. So the stain has a hard time getting into the wall to stain it. So those are your gram-negative bacteria. Does that make sense? Anything else, my friends? Don't stay up late. Listen to me. Listen to me. Pinky swear with somebody right now in the room. Pinky swear right now. Pinky swear. Just listen. You can unpinkify. You can unpinkify if you have to break it. Just repeat after me. Try. Here's the pinky swear. Listen up. Here is your pinky. Here is your pinky swear. Repeat after me. I will do my very best. To not procrastinate. And start reviewing. As soon as I possibly can. I will not be distracted. By anything on my phone. By anything on my computer. Or friends who bring me drama. I will be focused. I will have my lab ready. I will study. I will eat dinner. And if I don't exercise, I will go for a little walk. Because that is good for me. All right. Deal. Make good choices. Have a piece of toast.